All right, you guys. There are, and we're going to talk more about this in notes, there are 4 basic nitrogen bases in DNA. Guanine, cytosine, adenine, and thymine. Everybody say guanine is G. Cytosine is C. Adenine is A. And thymine is T. So we're going to learn more about the base pairing rules later, but G goes with C and A goes with T. So how am I going to know? You can see here how you have adenine with thymine and you have guanine with cytosine. Okay? This is the middle of the DNA molecule. So when you look at DNA, you're looking at monomers of nucleic acids called, anybody remember the monomers of nucleic acids? Everybody say nucleotides. Nucleotides. And a nucleotide has 3 basic parts. It has a phosphate group, which is the same in all nucleotides. It has a sugar, which is the same in all DNA nucleotides. It's called deoxyribose. And then it will have a nitrogen base in the middle. The nitrogen bases are held together by weak hydrogen bonds. Okay? So DNA, if it's going to break, is it going to break down the sides or down the middle? Down the middle, because there's weak what there? Hydrogen bonds. Okay? So when you unzip DNA to copy it, you unzip it down the middle. Think of it as a zipper on your jacket. Okay? You can unzip DNA into 2 sides. The 2 sides are made of these nitrogen bases, adenine pairing with thymine, guanine with cytosine. So if I know one side, do I automatically know the other? Yes. So if I know I have guanine here, I must have what over here? Cytosine, right? Okay? How do I remember this? Here's how I remember it. When I get vitamins, I go at G and C. A with T, G with C. Get that? No? Okay. That's how I remember, because you go, you know G and C, the store? G goes with C. Plus the G and the C both have what? Curves, right? It's kind of the same shape. The other ones are made of straight lines. You can remember it that way too. Here's the thing. Adenine and guanine are 2 types of what are called purines. Everybody say purines. Purines are bigger. They're 2 carbon structures, 2 ring carbon structures. We're going to take notes over this later. Don't freak out too much right now. Okay? So there's 2 types of purines, A and G. Adenine and guanine are my tier 2 purines. How am I going to remember that adenine and guanine are purines, guys? You want to know how I remember it? How do you kill a werewolf? Silver, which is what? A and G, like pure silver, right? Okay, purines. So that's how I remember it. Okay? Literally, every time I got to remember purines and purines, I go, I got to kill a werewolf with pure silver. So purines are AG, okay? And then, what? That's okay if you don't. Then you can just remember it. All right, that works too. And then the other ones are called pyrimidines. Pyrimidines are smaller. Oops, I got to move it up. So we understand that our two purines are G. Oops, sorry, I should do A and G. A and G. And my 2 pyrimidines are C and T. I don't have any special way to remember this one. Okay, I'll be honest. You could say, okay, I'm going to spell King Tut that lives in a pyramid. I do that sometimes in my head, but King spelled with a C, that doesn't work. But if that helps you remember it, go for it. Okay? But I always remember Tut, pyramid, pyrimidine. C is the left out one, right? Or cats in the pyramids. Tut, cat Tut, I don't know. Because, Egyptians, right? Egyptian, pyramids, cats, and King Tut. That works. Okay? What? What'd you call me? You try to... Yeah, but it's called pegging. It's actually a mental thing. So if you can peg things to other things, make more synapses, you're good to go. So you guys, you need to understand that there's two basic types of nucleotides. There's purines and pyrimidines. You always have to have a purine with a pyrimidine. And G goes with C and A goes with T. So you go at G and C to get your vitamins. That's how I remember that. I don't care how you remember it, but you have to remember. So the reason why is this. Purines are bigger than pyrimidines, right? Imagine two big ones next to each other. We'd have a bubble, right, in DNA? And then if we had two little ones, we'd have an indentation. This keeps everything balanced and equal. The side chains are the same. What is DNA made out of? Monomers called what? Nucleotides. Okay? The sides are held together by covalent bonds, the middle weak hydrogen bonds. Okay? So, in front of you on page 5, you have this basic color schematic. You have to decide what we're going to color all of these parts for the rest of the day. So, what color do you guys want to make the sugars? I heard blue, purple, what do you have? Yellow. Not yet. Not yet. Pink? Invisible? No. Blue I heard twice. I'm going to go with that. That doesn't really look blue, but we're going to say it is. That is not bad, is it? Oh, it's a highlight. Yeah, that's what it is. All right. All right. I done goofed again. Let's do this. Now I won't be able to see through it, though. Okay, so your sugars get colored blue. That is, it's a vibrant, that's royal blue. All right. The phosphates are going to be black. I can't change that. Okay? Phosphates are black. Adenine, thymine, guanine, and cytosine are each going to need their own color. I heard guanine green. Gee, that makes sense to me. Where's Christmas green? I don't see Christmas green. That. Okay, so guanine's green. You guys should be writing this down, though. Just next to it on there, it's fine. On this point, you can. Everybody the whole day is going to have the same color schematic. Because we're going to put our DNA together. We're going to have this long DNA strand when we're done. Okay? I need a color for the other pyrimidine. What? Red. Cytosine's going to be red. What color are we going to make thymine? I heard purple. Be careful, we got to have color marker for it. I heard yellow. You want yellow? T? Okay. Yeller. Old yeller. Old yeller. Hurry up and shoot him, right? Okay. Terrible movie. They made us watch that in 4th grade. I remember that. Watch a movie about killing a dog. It's terrible. Terrible. So, everybody got this? Okay. The next thing I need to do